Hello everyone, so this is a re-recording of the workshop given at Drug Discovery Africa on foundations of computational generative design of small molecules, construction, scoring and search. So this recording is actually broken up into several smaller videos, so you can pause, repeat, move around. And there is also the recording of the NIME component as well. So that is after the lecture material. So first of all, to jump in, thinking then about the learning outcomes. So our learning outcomes are first of all trying to understand basic chemical and reaction representations and how they can be used to design new molecules. So this is really foundational to be able to understand the molecular representations that are involved in generative design. And then it's moving on to the three pillars of small molecule generative design. So that is construction, scoring, and search. Then it's understanding molecular construction in more detail. So what are the pros and the cons of each? Then thinking about scoring, so explicit scoring, implicit scoring, receptor and ligand-based scoring. And then finally understanding deterministic and stochastic search. So hopefully these are all of the sort of things that we'll be able to understand as we move through this series. The things then I'm not going to talk about in this particular series is that I'm not going to talk about virtual screening or convincing chemists to make our molecules, nor issues around particular series. All the examples here are from the literature. Nor am I going to talk about building or validating different generative design methods, nor the engineering or requirement of HPC. And certainly I'm not going to be able to cover every new method that is described in the literature. There is certainly an absolute plethora of different approaches, so we can only focus on a sort of narrower subset and really look at the foundations of the field. So first of all, we're going to start with an introduction to generative design. And generative design is really all about trying to create novel molecules from scratch. And this is really used as part of what we call the design, make, test, analyze cycle, so the DMTA cycle. And this is where we will design molecules, have them made, tested, and then analyzed. And as an example, I show sort of in the bottom left a fragment which I have then used generative design to generate more sort of small molecules from. And what you can see is that these molecules are sort of very sort of different to the starting material, and I think it's interesting because actually these molecules, due to the size of chemical space, are unlikely to ever have been seen before. So it's kind of exciting that we can actually be able to use these tools to design new novel molecules which have the properties that we want. And this really then moves our mindset in drug discovery from what compounds do we have to screen to what compounds should we make next? How do we actually go about traversing the depths of chemical space? So then when we start looking at sort of the depths of chemical space, generative design really helps us go out and explore and overcome the current limitations that we have with existing conventional sort of chemical libraries. We can start to really explore novel chemical space. We can create compounds with the properties that we want, and we can also overcome intellectual property issues to be able to find novel structures. And this really allows us to be able to try to address challenging targets where existing approaches have possibly failed in the past. One of the interesting sort of papers that explores this is where the sort of edges of chemical space and really pushing generative design to the limit, these authors went away and tried to explore the depths of chemical space. And what they found was that actually when you start to look out into the depths, we find that the majority of chemical space has been underexplored. So everywhere that you see a color on this particular plot, that's somewhere that we have made a compound, in this case from PubChem, and you can see that there is an awful lot of white areas which are unexplored chemical space. Now, one of the sort of then questions is what do these sort of unexplored areas contain? And we can see that they contain very complex structures, and that sort of highlights the fact that when we're thinking about generative design in what chemical space can we access, we can see that we can explore a significantly more complex space than perhaps we can with traditional sort of design thinking. I think it's then important then to kind of shift gears a little bit and start to then think about, okay, what are the key components to be able to build generative design software? And there's really sort of three key pillars, and beneath that is a sort of foundation in terms of cheminformatics. So the foundations is certainly in representations, and each of the pillars then are molecule construction, molecule scoring, and molecule search. So molecule construction, how do we build molecules? How do we then a molecule scoring, how do we score molecules? And then how do we search and traverse the space successfully? When we then thinking about that sort of question, it ties back a little bit to the example of traversing deep chemical space. Another aspect that we have to keep in mind is that chemical space is astronomically large, of course, but it's also highly discontinuous. That is, there are plenty of gaps inside the chemical space. There are areas which we may be able to design in terms of on a computer, we can draw a depiction of that molecule. However, in practice, we may never be able to synthesize them. It may not physically be possible to make that type of molecule. And what that means is that we end up with gaps in the chemical space. And gaps in the chemical space are really areas in the chemical space where we can't physically make that molecule, or that molecule cannot physically exist in reality. So whilst it might be sort of on the surface straightforward to be able to identify a particular chemical graph, it may not actually be possible to make that in practice.